And the thing that's interesting about that for us is that recently Frank McCune's lab has put forth a very provocative model for how you might get cancer in different tissues. And so this comes from his work on Barrett's esophagus, where you've probably all heard of acid reflux. That's pretty common. And some people have that long enough that they feel a little lump in their throat. And they actually are lucky because they'll go into the doctor and they'll get scoped. And when they look in your throat, they'll see a little pile of cells in there that don't look like they belong to the esophagus at all. And that is Barrett's esophagus. It's a precancerous lesion. And so people are usually treated to prevent that, because if it's left untreated, you can go on to get esophageal cancer. So here's the esophagus. Here is the stomach. And the McCune group identified a group of quiescent cells that had not been discovered before at the junction between these two tissues. And in their model, continuous acid reflux damages the cells that are lining the esophagus, and that in response to that damage, these quiescent cells actually lose quiescence, proliferate, and regenerate the tissue, but they do it inappropriately. And the epithelial cells that are made here, the esophagus is normally squamous epithelium, and these cells actually make something that closely resembles the intestinal epithelium, so it's not the right kind of cells. And so we think that the parallels between seeing our quiescent population lose quiescence in response to damage is actually quite provocative, and we think that understanding how you regulate quiescence entry and exit from that in the context of stem cells and damage is very interesting and will be very useful for more than just flies. So we very much want to know what signals change in the niche when we've damaged it like this, because we think that'll teach us some very interesting basic biology about both normal and damaged niches. So, now you guys can ask me anything you want. Because, yeah?
On the Barrett slide that we just left,
Mm-hmm.
you said the residual embryonic cells?
Yes.
I always thought embryonic stem cells are only in an embryo.
What are embryonic stem cells? Embryonic stem cells are derived from the inner cell mass of a blastocyst, and so they are, I think Rudy Yanish had this nice quote, a tissue culture artifact, although a very useful one. Okay, so keep that in mind, that in the normal embryo there are no such thing as an embryonic stem cell, but when you plate them out and grow them in culture, that's when you get an embryonic stem cell line. And now, fast forward to the present, and people are deriving very similar pluripotent cells in culture, induced pluripotent stem cells first from fibroblasts, but now from many, many tissues. So embryonic stem cells are cells that can be grown in a dish, but we are actually talking here about, you would call it an adult stem cell, the stem cell that regenerates the esophagus normally. Yeah, those are the two very broad categories of stem cells, so that's a great question, and thanks for asking about that.
So the green cells are actually adult stem cells?
They are not even stem cells. They are normally quiescent cells, and upon damage, lose quiescence, proliferate. Whether they're a stem cell or a progenitor cell is definitely not known. We didn't talk about how you identify a stem cell, but there are two major ways. There's a functional assay, hematopoietic stem cells being the gold standard, where you can transplant a patient and reconstitute their whole blood lineage if you have a hematopoietic stem cell in the mix. And the way that you study it in an adult tissue is to do lineage tracing, where you mark the cell genetically and permanently and follow its behavior over time. So those are the ways that you actually identify stem cells. Yeah?
You know, when you ablate the somatic cells and reduce it back to the hub, is there any, again, my understanding is most of it is signaling from the environment, but is there any role for mechanical forces?
Mechanical forces, role for mechanical forces in tissue regeneration. It's got to be important, because all the cell biology now is showing us, you can take cultured cells and change their cell fate choices by just changing mechanical forces. It's much, much harder to study mechanical forces in an intact tissue like this, but I am sure they play some kind of role. It's just very, very hard to test. I am using signaling in a very vague sense, and from my point of view as a cell biologist, you have to think about every single cell as a beacon of signals at every moment in time, always, and all you can do is focus on the ones you can understand the function of. So the ones we understand best are these hub cells, which are like a beacon of a local set of cytokines. And so what we'll first go after is looking at how is the hub different in testes that are normally functioning and in testes that are doing this massive regeneration, because we think, but have not even proven yet, that those signals will change. So that's one thing. And then, yes, we very much are curious to know whether or not damage introduces a whole new set of signals that would normally not be there because of a secondary consequence of having a bunch of dead stuff in the tissue, for instance. That's known to change signaling. We also think that, yes, normally there must be breaks signaling to the hub to keep it from losing quiescence all the time, and that the cells that we've removed, these somatic stem cells, maybe they are actively preventing hub cells from losing quiescence. And again, this is not very well understood in any tissues right now. So that's why this is great to be able to study this with a set of cells that we can manipulate signaling in genetically and figure out what's happening. But I'm not going to pretend like we're going to answer that quickly. That will take us years to figure out. Yeah?
Can I ask you a rookie question? If you could back up, did I understand you that there are no embryonic stems, there are no stem cells in the embryo, there are only stem cells when you take them out and plate them? I'm not sure about that.
So there are two broad categories of stem cells, embryonic and adult. And there are many, many cells from different parts of the embryo that can be plated out in culture and pushed down to a pluripotent state that will then differentiate into all different kinds of cells depending on how you culture them. But those are not cells that would have done that normally in an embryo. They have been changed in culture, and they're a very useful tissue source, but they are not analogous to any endogenous cells within an embryo. And then to the point of, are there stem cells in an embryo? Actually, I like what Irving Weissman, the president of the ISSCR, has stated very clearly, which is stem cells are in adult tissues. You know, they are what are regenerating the adult tissues. So that's a very sticky term. It's always good to just communicate with whoever you're talking with and decide if you're working with the same definition of these things. Because in a lot of cases, people will say stem/progenitor cell because they're not sure which it is. And to people like me, that drives me crazy because we know that the stem cell and the daughter cell that you would call a progenitor have very different fates, right? But in most cases, it's very hard to distinguish them. So the words matter, actually. So that's where we need writers. Yeah?
Do all stem cell niches have hubs like this?
So that is one of the very big overarching questions. There are many cases where there are quiescent signaling cells, for instance in the hair follicle, that are definitely critical for regenerating hair and skin. I showed you the mammalian testis, and it's not clear which cells might be the analog of the hub, but that is what people are looking for in all kinds of situations. And in some cases, it might not even be a group of cells, it might be a different extracellular matrix environment. So we're just lucky that we can see these cells and manipulate them. But the concept is that, yes, many tissues will have something analogous to this.